Proteins are unbranched and linear polymers called polypeptides, and they're composed of amino acid monomers. Every amino acid shares the same general structure. This contains an amino, carboxyl, and R group connected together through a carbon atom with a hydrogen atom. The R group can vary, and this is what distinguishes different amino acids. Most R groups can be classified into categories. Electrically charged R groups will attract water and opposite charges. Polar R groups will form hydrogen bonds with water and other polar or charged substances. Non-polar R groups will bunch together away from water in the protein's interior. The proteins in the human body are made up of combinations of 20 amino acids. Out of 20, we have 9 essential amino acids, which we can't naturally produce and have to gain by food consumption. Through a condensation reaction, the hydroxyl in the carboxyl and hydrogen in the amino group combine to form a water molecule. This creates a covalent peptide bond in what is now called a dipeptide molecule. Multiple condensation reactions between the amino acids will create a polypeptide chain. The shape of a protein is made up of 4 levels of structure. The primary structure is a sequence of amino acids. This is controlled by DNA coding for protein synthesis. Primary structure ultimately determines the shape of a protein, and therefore its function. The secondary structure is the curling or folding of a polypeptide chain into alpha helices or beta pleated sheets due to the formation of hydrogen bonds. Hydrogen bonds form between the slightly negative oxygen in the carboxyl group and the slightly positive hydrogen in the amino group. These hydrogen bonds are weak individually, but they're strong collectively. The tertiary structure is the overall specific 3-dimensional shape of a protein. This occurs through the interactions between R groups and their different properties. Hydrogen bonds are formed between polar R groups. This is the same type of bond found in the secondary structure, and this stabilizes the folds in the protein. Covalent disulfide bridges are formed between sulfur atoms in the R group of a cysteine amino acid. These bonds are strong and are not easily broken by changes in the environment. Ionic bonds are formed between positive and negative charges in the R groups. These bonds are weaker than the disulfide bonds. However, similar to the hydrogen bonds, when the temperature is increased, this will cause the molecules to move more rapidly and break the ionic bonds. When the pH is altered outside of its optimal, ionic attractions and repulsions in the R groups are changed. Interactions between hydrophobic side chains cause them to aggregate together in the interior of a protein, which causes folding that is stabilized by van der Waals forces. From a quote from Sadava in 2006, a complete description of a protein's tertiary structure specifies the location of every atom in the molecule in a 3-dimensional space in relation to all the other atoms. The quaternary structure is the specific 3-dimensional shape of a protein, determined by multiple polypeptide chains, which are called subunits, and or prosthetic groups bonded together. Prosthetic groups are non-protein components which are necessary for a protein to function. The quaternary structure is actually not rigid and can slightly shift its subunits to perform its function better. Globular proteins are proteins with a spherical shape. They're normally soluble in water, and they play metabolic roles. Its specific shape allows for specific functions. Fibrous proteins are long and strong proteins. They have structural roles since they're made up of long polypeptide chains with non-polar R groups. This forms fibers which are insoluble in water. The structure of a protein directly relates to its function. A protein's specific shape allows it to bind non-covalently to another molecule. Carrier proteins are used in membrane transport as they have a specific binding site to allow substances into and out of the cell. Chemical signals can bind to receptor proteins, so they are used in signaling. Enzymes have an active site complementary to a substrate. They are biological catalysts which speed up chemical reactions. Antibodies bind to antigens on the surface of pathogens and cause a series of immune responses. When a protein is denatured, the bonds in the secondary and tertiary structure are broken, which destroys the protein's biological function, since this causes a new structure to form or different molecules to bind to the protein.